Okay, so this is your recorded lecture for April 14th. So please make sure well, I hope you'll be listening to this before April 21st. Okay, so I'll continue where we left off last time. And like usual, these are some of recap questions, which also helps you study and know what to try to remember. So for legume and oilseed proteins, if we say what are the main protein classes, so we have the globulins and we have the albumins, and then the globulins are mostly the storage proteins that are contribute to functionality of the protein, whereas albumins are mostly enzymes and structural proteins, and of lower molecular weight, smaller proteins, and mostly monomers compared to the globulin proteins. So when we ask about what are the molecular differences among the different classes, so we want to remember, okay, so what are some differences in the globulins? Let's see here. Yeah, so the globulin proteins specifically, we have two fractions, the 7S and the 11S. So the S, again, we have to remember that this is related to the sedimentation coefficient, which is correlated with the molecular size of these fractions. So the larger the molecular size, that means the higher is the velocity at which they sediment at a particular gravitational force. So 11S is larger than 7S, larger in terms of molecular weight and size. And the 11S forms a hexamer, and the hexamer is made up of two trimers associated together by hydrogen bonding and hydrophilic interactions. And in each trimer, you have three monomers. Each monomer is made up of the acidic and the basic subunits. So the acidic are the black spheres and the basic are the blue spheres, and they are associated via disulfide linkages. And the blue sphere is of a lower molecular weight, which is the basic subunit, is of lower molecular weight than the acidic subunit. And then each of these monomers, so the monomers in a trimer are linked by hydrophobic interactions. So in terms of the 7S, it is a trimer made up of three monomers, and they are linked together via hydrophobic interactions. So the 7S and 11S are the main globulin fractions in legumes and oilseeds. So you have 7S and 11S in soy, in pea, for example, in chickpea. So you have these fractions, but they differ in amino acid composition and sequence. And one thing we said, for example, in pea, there is another 8S fraction that is in pea that is called convicillin. So the 11S is legumin, and the 7S is vicillin. And in soy specifically, so when we say what are the soy protein subunits, so there are specifically glycinin as the 11S and beta-conglycinin as the 7S. And here is a gel with the three monomers of the beta-conglycinin, alpha prime, alpha, and beta subunits. And the glycinin, we have the acidic banding and the basic subunit. So the acidic, we have different species. So we have different acidic varieties or species that are known to exist, or different variants, basically. So this is a gel run under reducing conditions. Otherwise, Under non-reducing conditions, the acidic and basic subunits will be linked together, and then they won't migrate into individual bands. Another reminder is the effect of pH on solubility. So under acidic conditions, the protein is moderately soluble around the isoelectric point. It loses solubility because then you lose net charge, and then the protein is governed by hydrophobic interactions, so it falls out of solution. And at alkaline pH, the protein is more soluble than acidic due to ionization of other groups, such as the OH groups in tyrosine, and then sulfurilyl groups in cysteine are get ionized, and you have higher solubility under alkaline pH. Okay, so this is kind of a recap, a little bit of a recap, and then we're going to move on and talk more specifically about solubility of the different fractions of soy. In this example here, this is actually an article that I posted under reading material. I think it's week 11 reading material or week 10, you guys have to check. And there are 3 articles that would help as part of the studying material related to what I'm going to cover next here. This is an isolated fraction, glycine, isolated from soy. And here we're looking at the impact of different ionic strengths on the solubility curve of glycine. So we see something here very interesting. Like if you see at ionic strength of 0.5, so higher salt or ion concentration, what we see is this unique curve. It starts with very low solubility here, and the solubility increases as we increase the pH. Now, what this research has looked into, it determined that the acidic subunits within glycine dominates the exterior of the monomer or the protein 12 times more than the basic subunits. So basically, your acidic subunits with the additional carboxyl group is dominating on the surface. So at low pH, what happens is at low pH is your carboxyl groups get protonated. So you lose the charge, and you don't have much of a basic positive charge subunits to add to the charge of the protein. And with the high ionic strength, so there's a competition between the water interacting with the salt versus the protein, and then the salt can shield some of the charges on that, on the protein. So you see very low solubility at acidic pH. As the pH increases at this ionic strength of 0.5, what happens is then your carboxyl groups start giving out their protons. So we start gaining negative charges. And with gaining negative charges, solubility will increase. Okay, if we look at low ionic strength, at low ionic strength concentration, these 2 lines here are different ionic strengths, but relatively lower compared to the 0.5 ionic strength. So what happens here when you have low ionic strength, then the basic groups are exposed more to the surface. So instead of having 12 times more acidic groups on the surface, it becomes 4 times more acidic compared to basic group on the surface. So we have some basic groups, so we have some positive charges. So we have positive charges at the acidic pH. So the carboxyl groups get protonated, and the amino groups of the basic subunits are positively charged, so you have more charge on the protein, and then you have less salt, so you have more water interacting with the protein. So we see good solubility at these acidic conditions. Now, at this low ionic strength, and when we decrease the pH, what happens is we'll start to lose charge on the surface, and then the protein loses solubility. But then as we increase the pH again, then we start getting that negative charge and more interaction with water at this low ionic strength. So that's why we see two different types of curves, one that starts low at low pH and increases, and one that starts high at low pH, decreases around the isoelectric point and increases. So this is just to illustrate that salt content, pH content, the pH level, both impact the solubility of these fractions, specifically in this case is glycine. Okay, so this is another article that looked specifically at the solubility of fractionated acidic and basic subunits. So it's not just the glycine. So here we look at three different curves. One is for the basic subunits, this one at the bottom for basic subunit. And then for the acidic is the dark black squares. And then the glycine fraction, which is a combination of acidic and basic subunits. So what we see here is we have this the acidic subunits follow similar trend to the entire glycine fraction, whereas the basic subunit here has this very low solubility and much wider range of pH at which it has very low solubility. So it's the isoelectric point is very, very wide. Now, why is that? So if we look at the molecular level as to why is this, let's look at the amino acid composition. So this is the column for acidic subunits, and this is the column for basic subunits. So the amino acid composition, if we want to compare these four amino acid, alanine, valine, leucine, and phenylalanine. These are very hydrophobic amino acid, and presence of these amino acid at a particularly high percentage makes the protein the protein becomes governed by hydrophobic interaction, largely governed by hydrophobic interaction. So if we look at the percentages, so we have higher percentage of hydrophobic amino acid in the basic subunit compared to the acidic subunits. So in general, all of the, taking into account all of the hydrophobic amino acid, we have 47% hydrophobic amino acid in the basic versus 36% hydrophobic amino acid in the acidic subunits. But if we look at these four in particular, are the main ones that are different. So in the basic subunits, we have 28% of the amino acids are valine, leucine, isoleucine, and phenylalanine, whereas in the acidic, it's 20%. So 28% is very high concentration of these uniquely hydrophobic amino acids. So over a wide range of pH, the level of hydrophobicity dominates any presence of charge. So basically, this high level of hydrophobicity causes more protein-protein interactions and less protein-water interaction. So, and then when we decrease the pH further or increase it further, we enhance the solubility a little bit, but not by a lot because of the dominancy of these amino acids, the hydrophobic amino acids. In the acidic subunits, because of this percentage, overall the percent hydrophobicity is lower, and these specific four amino acids are much lower. So you have better solubility at acidic and alkaline pH and a narrower U shape. That means the isoelectric point falls in a narrower range than the basic subunits. If we look at glycine, the curve simulates more the acidic. It gets a little bit wider because it has the basic subunits as well, but mostly it resembles the acidic subunits. And here's where I usually would ask you, why are they the same? So, think about it for a few seconds. Why the glycine subunits or the glycine fraction resembles the acidic subunit curve. Okay. I know nobody's going to answer me right now, but I'm giving you a moment to think. But if you remember what I said earlier, is that at a particular ionic strength, even at low ionic strength, on the surface of the protein, you have dominancy of the acidic subunits. So acidic subunits are mostly dominating the surface, where the basic subunits are mostly in the core due to the high percentage of their hydrophobic groups. So therefore, because the surface of the protein is mostly dominated by the acidic subunits, then the solubility of that protein will basically simulate the solubility of the acidic subunits, because most in that fraction of the glycinin, the acidic subunits are mostly on the surface. Okay, so that is the reason why they resemble the glycinin fraction resembles the solubility of the acidic subunits within this fraction. Okay, so this is another article that looked at solubility of beta-glycinin in this case, and differences among the species and whether or not they're homotrimers or heterotrimers. To remember, when I say homotrimers, basically it's a trimer. Beta-glycinin is a trimer. That means 3 monomers linked together by hydrophobic interactions. And when we say homotrimers, then if we say we have, if you remember, we have alpha, alpha prime, and beta conglycinins. So the homotrimers, meaning all 3 of them are either alpha, or all 3 of them are alpha prime, or all 3 of them are beta. So that's what homotrimer and heterotrimer is, that you have different monomers within in one trimer, basically. So here you don't need to remember any of this. It's just showing you differences among between homotrimers and heterotrimers at different pH and different ionic strength, but you really don't have to remember any of these numbers. Okay, so the functionality of soy protein, or in particular, well, I'm talking about legumes and oilseeds, but we talk a lot about soy because it's the main legume protein in the market, really, with now pea gaining traction as well. But in terms of what is soy protein ingredient used for, it's not really used for its solubility. This is not that. It doesn't have that great of a solubility at acidic pHs, especially if you're looking at acidic beverages. It's not the first choice to go for an acidic beverage because of its low solubility. And around pH 7, it's soluble, it's not that great, but yet. So it's mostly used for its gelation and emulsification capabilities. Not so much for elasticity and viscosity, but the main functionality of the protein is emulsion and gelation. So here's just a quick reminder about gelation. We have to remember that the gelation, or thermally induced gelation, is dependent on the heat that we apply, whether it is the heating temperature and heating rate, as well as protein concentration. What is the critical protein concentration that a gel is formed at, and then the pH of the system and the ionic strength. So you don't want extreme pHs, and you don't want pHs at the isoelectric point, because then you have gritty gels, more of aggregates rather than balanced 3-dimensional gel. And sometimes you increase the salt concentration to help with formation of the protein-protein interactions, and sometimes a little bit of salt also helps protein-water interaction. So all of these factors are important. So if we're looking at soy, this is the same. The glycinin, for example, denatures at a little bit over 90 degrees Celsius. So you want to heat above the denaturation temperature, because if you heat below the denaturation temperature, then you end up with, you might form a gel, but it's reversible because it is, you don't induce really denaturation, so you don't have disulfide interchange, and potentially it's only hydrophilic, ionic and hydrogen bonding type of interaction. So it is reversible. So what happens is that at pH 7, at a particular ionic strength, a little bit over 100, we form our soluble aggregates. So if the concentration of the protein is lower than a critical concentration that forms a gel, these soluble aggregates will remain in solution and you don't really form a gel. So you want to have specific protein concentration that allows the formation of a network. And then if we look at soy, for example, it can form a gel around 10%. It can form a good gel around 15% protein concentration. Beta-conglycine, if we look at the fraction separate from glycine, beta-conglycine does not form a strong gel because we don't have disulfide interchange. It's very low in sulfur-containing amino acids, does not form disulfide linkages. So interactions are slightly weaker than it would be if you have glycine fractions. So, and also beta-conglycine is glycosylated, so more charge on the surface, high acidity, that means high net negative charge, and therefore the gels are not very strong gels. Obviously, it will be nice to have a mixed system. Obviously, well, you don't need to fractionate glycine and beta-conglycine separately. In soybean, they're present together, but a good ratio between the two allows for a good gel formation. And it is definitely dependent on the ratio and also dependent on the types of the subunits present or the species of the subunits present. So here's a research article that looked at the impact of the different subunits, there's different components, fractions on the gelation, and they used enzymes to determine the impact of the different fractions or different components of the protein on gelation. So what you see here is, so we have the soy protein in this lane 2, soy protein, actually lane 4 is the soy protein isolate, where you have the beta-conglycine and glycine. And then in lane 3, this is the isolate, but has been hydrolyzed by pepsin. Pepsin targeted glycine subunits, and you see that they're no longer there, the glycine subunits, but you see smaller peptides here. Whereas another type of enzyme, which is the papain, it targets the hydrolysis of beta-conglycine. So you see those bands are not there anymore, and you see that the glycine subunits are intact. So they took these hydrolysates and tried to form a gel. They noted that the reduced conglycine hydrolysate. So R is for reduced, C is for conglycine, and H for hydrolysate. So the reduced conglycine hydrolysate had slightly higher gel strength or hardness than the control. And the reduced glycine hydrolysate, that means the hydrolysis that we broke down the glycine components into small molecular weight, we see a dramatic decrease in the gelation strength or the gel hardness. So this indicates that when you break down glycine into smaller peptides, then you lose that ability to form a strong gel. Whereas when you break down beta-conglycine, you don't see much of an impact on the gel formation, maybe slight improvement because, you know, I don't know if it's even statistically significant, but it didn't impact, I would say, the gel formation as much as having a lower glycine content. So that emphasizes the importance of the ratio of glycine to beta-conglycine. So in terms of emulsification properties, again, the ratio of glycine to beta-conglycine is important. Glycine here plays more of a significant role than glycine in the formation of an emulsion because of its smaller molecular weight. So in general, it moves to the interface faster and easier. It doesn't have disulfide linkages, so it's more flexible at the interface. And also it has a better balance between hydrophobic and hydrophilic groups. So it has higher surface hydrophobicity, but at the same time, it has higher net surface charge because of the presence of glycated amino acid and also higher levels of acidic amino acids. So overall, it has a good balance of hydrophilic, hydrophobic residues on the surface. So if we want to compare different legumes, if we're looking in this case at soy versus pea, so they're both legumes and pea is being now is getting more attention and is used more in the market than a few years back. But if we look at the functionality here, there is a great difference between the functionality of soy protein isolate and pea protein isolate. So in this particular pea protein isolate, it is formed at 20% protein, and this one is formed at 15% protein, but still it shows much higher, this is rupture force, that means much higher gel strength. In terms of emulsification, also the soy isolate has much better emulsification properties than pea in this case. Now, of course, there is an impact of processing, how they were produced. Soy has several decades of processing advantage compared to pea. So it's processed in a way that either enhances its functionality or preserves its functional properties. Another important difference here is we have a higher legumin to viscellin ratio. So in soy, we have more of legumin than viscellin, or the higher ratio of legumin to viscellin. In pea, you have lower ratio of legumin to viscellin, and that could impart differences in functionality. So different protein ingredients out there, we talked about that a little bit a little bit about different ingredients at the beginning and the initial first lecture, but here's just a reminder. So if we look at soy, we have the flour, which is really high in protein, 50% protein in defatted flour. And then we have the concentrate that have about 65 to 70% protein. And we have the isolate that at least have 90% protein, if not higher. So they're protein-rich ingredient compared, like, for example, if you talk about pea flour, it has only about 20 to 25% protein compared to soy flour that has 50% protein. And then pea protein concentrate is about 55% protein compared to up to 70% protein in soy protein concentrate. Okay, so uses, there are so many different uses for soy ingredients, flour and concentrate and isolates, but the isolate is the most widely used given in different food application, given its cleanest flavor. It has the least beany flavor and has, because of the highest protein content, the most protein functionality. But soy flour and soy concentrate are also used in bakery products, in meat products for extrusion, so they are also used. Okay, so this is kind of an overview of processing of the soybean. So obviously it's a legume, so it comes in the pod, and then it requires dehulling because of the outer hull, kind of, it's mostly fiber and doesn't have the, of course, the oil, and the protein is in seed, not in the hull. So first of all, you get the soybean, do cleaning and cracking, and then you get the hulls, and then you have the soy chips. Soy chips goes through conditioning by heat and flaking, and then you get the full fat flakes. The full fat flakes get extracted for oil, so you get the oil removed. There's the pressing, and then there's also defatting by solvent to remove most, if not all, most of the oil out. And because you used hexane, they do desolvenitization step in industry. It requires heat for higher efficiency of time and removing of that solvent. And then you get the defatted flakes. Defatted flakes can go multiple routes, either directly for protein extraction, or they can go through further grinding, and you get the soy grits, and then further grinding to finer particles, you get the flour. So at the point of defatted flakes, there's two routes to produce the two different ingredients. Removal of sugar, that could be done with either alcohol or acidification. Mostly in industry, they go with acidification to avoid the use of alcohol. And it's basically precipitating dispersing the protein, the flakes in water, and then reducing the pH to the isoelectric point of the protein. Then they remove the solubles, and then the precipitate has some fiber and protein, and dry, pasteurize and dry by spray drying, and you get a concentrate. If you want to get an isolate, you go through the route of protein solubilization under alkaline conditions. Then you remove the precipitate, which is mostly fiber, and then you get the supernatant, and then you subject it to an isoelectric point pH, precipitate the protein, remove the solubles, sugars and fiber, soluble fiber in this case, and then spray dry, neutralize first, spray dry, and you end up with an isolate. There's more than 90% protein. So these are different ingredients, and the concentrates and the flour can be used for extrusion, where you have the oil entry, water entry, blending, texturizing, drying, packaging, etc. This is the another breakdown of how you produce the concentrate and isolates. And then here, for the processing of soy, you can produce soy milk, which could be a starting material for tofu here. You get the soybeans, they're soaked, they're ground with water. You get the slurry, gets heated here. It's important to heat as a kill step and also in activating of trypsin inhibitor. And then filtration, you get the okara or the residue here, and then you get soy milk. Soy milk here, about 5 to 6% solid, so it's below the critical concentration of forming a gel. So you heat it up to denature some of that protein, and then you add a coagulant to help formation of gel at low solid content. So in this case, it's calcium ions are added, and then pressed to remove more moisture, and you get soy milk whey, and then you get the curd that gets cooled and cut, and you get your regular tofu. This is the texturization route, where you get the flour or the concentrate mixed with water, other ingredient, and extruded obviously under pressure. And the extrudate has this unique fibrous texture under certain conditions. Again, can be cut into different shapes, and it's another meat-like product. So soy proteins specifically are used a lot, not just for their functional contribution, but also for nutritional benefits. And we talked about that also at the beginning of the semester, where soy has a PDCAS of 1 comparable to animal protein, and all of the rest of plant proteins are lagging behind in terms of PDCAS. Also, it has been shown that it's linked to prevention of cardiovascular diseases through reduction of saturated fat and cholesterol, and has other health benefits, decreased risk of breast cancer and other cancer, prevent osteoporosis, and a lot of the benefits are associated with the phytochemicals known as isoflavones that are abundant in soy. Okay, so this concludes the legumes lecture. And I am going to go ahead and start the cereal protein lecture. So if you want to take a break, you can pause the recording. I'm just going to continue here into cereal proteins and hopefully finish that up. It's not a long lecture, so I'll be able to finish it in 50 minutes. So here we go. So cereal proteins now next. So far we have covered dairy protein, legumes, and oilseeds, and now we're moving on to cereal proteins. So this is for you to think about. So oftentimes at mills where they produce the flour, they add ascorbic acid. They fortify the flour with ascorbic acid. And this is for either the addition of ascorbic acid, the gluten strength increases upon kneading, and elasticity of the dough decreases and viscosity increases, and or disulfide interchange and intermolecular linkages increase. So if you want to think about the ascorbic acid, so ascorbic acid mixed with water becomes kind of oxidizing agent. So in the presence of oxygen, so the ascorbic acid oxidizes and induces disulfide linkages. So basically, when we think about kneading the flour with water, so what happens there is the protein is denatured and due to physical handling, and the protein or gluten-forming proteins start interacting with each other. In the presence of ascorbic acid, it facilitates disulfide interchange. So it oxidizes the sulfidyls into disulfide or into disulfide bond, and it increases the strength of gluten over the dough. So then we have enhanced elasticity. So B is incorrect. So elasticity of the dough increases and viscosity decreases in the presence of ascorbic acid. So, and then disulfide interchange and intermolecular linkages are enhanced in the presence of ascorbic acid. Okay. Usually I ask students at this point, is there gluten in wheat? Or before at the beginning of the lecture, I would say, is there gluten in wheat? And all of you would say yes, and I would tell you no. What's in wheat is gluten-forming proteins. So there is no gluten in wheat. Gluten is formed upon formation of the dough. Gluten is the network of the protein that interact with each other to form that viscoelastic structure, which is called gluten. Glass transition. And I would, at this point, I would ask you, what is glass transition? And you would think about it a little bit, and some of you might know, some of you have forgotten. It is the temperature at which the material changes from a glassy state to amorphous state. Glass transition of gluten can occur at room temperature under dry condition, at room temperature under moist condition, upon heating under low moisture condition. So what do you think is the answer? It does not form at room temperature under dry condition. There should be, the water facilitates glass transition at lower temperature. So you want some moisture, some amount of water to facilitate the conversion from a glassy state to amorphous state, a more flowy state, in order for the protein to become flexible and form the gluten structure. So it can happen for gluten at room temperature under certain moisture condition. But for example, for zein, which is corn protein, it has to be at higher temperature. It has higher glass transition temperature at same moisture content. So we can induce the glass transition to happen at lower temperature, at lower moisture conditions, but if we heat. So basically the moisture amount influences the temperature at which we have glass transition. So the answer to this is B and C. This is just FYI, the family of family, subfamily, tribe, and genus of different cereal grains. We're going to talk a lot about wheat. We're going to talk a little bit about corn, and not talk much about the rest. So in terms of protein content in cereal is not that great. You don't have that much of protein in the flour. It ranges between 7 and 14, 15%. Depending on the variety, you might get a little higher than that, but it won't exceed, in normal situation, 20%. If we look at wheat, for example, it says 12%, but this is really an average, because you have soft wheat, for example, varieties that have as low as 9% protein. And then you have the hard wheat somewhere between 11 to 13. And then you have the durum wheat that has 15, 16% protein. So different varieties, different protein content, really. So here's the breakdown of the different protein fractions in different cereal. So the main protein components, of course, you have albumins, but albumins are present at low concentration and they're not storage proteins. And then you have the globulins, and then you have prolamins. So the globulins, you have 7S and 11S. Similar to legumes and oilseeds. So you have those main fractions. If we look at wheat, for example, the globulin in wheat is trichitin, and it's present at small concentrations, so it's not very significant component of wheat. And you have other globulins in oats and rice. But prolamin is the major component in cereal. And you have prolamins, and you have a high molecular weight prolamins. For example, in wheat, the glutenins are glutellins. So they are under the big umbrella of prolamins, but they are the high molecular weight prolamins that associate to form bigger polymeric proteins. And they are specifically known as glutellins. So gliadin in wheat is the prolamin, the regular prolamin, and the glutenin is the high molecular weight prolamine that is known as gluten. So in oats, for example, your prolamine is called evenin. In maize, you have the zein protein, so beta-zein, gamma-zein. You also have the alpha-zein, which is the main prolamine constituent in corn or maize. In sorghum, you have the catherine, in rice, secalin, in barley, hordine. So these are the different prolamins in the different cereals. Of course, you don't have to remember the names, but if you see them in literature, now you know what they are. So in wheat specifically, prolamine proteins predominates. Okay, so that's mostly prolamine. So whether they are the regular prolamine or the high molecular weight prolamine, but these are the ones that predominate in wheat. Trictins are present, like I said, these are the globulins present in minor amount. So this particular protein in wheat, the prolamine and the high molecular weight prolamins, have very characteristic functionality that is very unique to wheat. I've yet to see another grain or seed that has such functionality, which is the viscoelastic properties that are very unique for this type of protein, for wheat protein. So in wheat, we have the classification of hard wheat, durum, and soft. So basically, they are characteristic is related to the protein content. So the durum has the highest protein content. On average 13, it can be like up to 15, 16, depending on the variety, and also hard wheat ranges between 10 to sometimes 13%. So these are just averages that you see here, the numbers. And the soft wheat contain lesser protein, but they also differ not only in the amount of protein, they also differ in their profile. So that means the ratio of prolamine to high molecular weight prolamine, or in wheat specifically, a gliadin to glutamine ratio. So hard wheat has higher amount of glutenins, and soft wheat has higher amount of gliadin. So in soft wheat, gliadin to glutenin ratio is lower than in hard wheat. There's also another characteristic that is linked to the hardness, is the amount of friabilin or pyrrondilins. So these are, for short, they call pin A and pin B, which are a specific type of endosperm proteins that they associate with the starch granule. And if they are present, this is characteristic for soft wheat. And if these are genetically not present, these are characteristic of hard wheat. You might come across that in literature if you're reading it, so that you have an idea what pin A and pin B are. So distribution of proteins versus in the bran versus germ versus the endosperm. So you have about 15% protein in the bran, which contributes to about 20% protein of the entire seed or kernel. And in the germ, it has 26% protein, which contributes to about 8% of the total kernel protein, but the most amount of protein is in the endosperm, which is a starchy component, but it does have about 10% protein, and that amount represents about 72% of the total kernel protein. Again, what are they? We have albumins, like I said, which are non-structural, non-storage proteins, and they are water soluble according to Osborne fractionation. So albumins are water soluble, globulin are soluble in salt solutions, prolamins are soluble in alcohol, and glutelin, which are the high molecular weight prolamins, are soluble in acidic, alkali, detergent, also in the presence of reducing agent, we can solubilize the glutenins. So albumins mostly and some globulins are, in this case, in the cereal world, in the wheat, in wheat grains or kernels, they are mostly cytoplasmic, membrane proteins, enzymes. They're not important for germinations or, well, important for germination, I'm sorry, and some enzyme inhibitors. So albumins and globulins in wheat are mostly in the bran and germ. So when we have whole wheat, you have an appreciable amount of albumin and globulin versus the bran-free wheat or white wheat flour. Refined, I'm searching for the word refined. Okay, so prolamins and high molecular weight prolamins. So in wheat, prolamin and glutelins, these are glidins and glutenins. So they're not gluten, there are gluten-forming proteins. So these are the proteins that contribute to the formation to the gluten network in doughs. So these are storage proteins used as sources of amino acid and nitrogen during germination. Some of the glutenins, due to the high molecular weight polymeric proteins, they can be referred to as structural proteins, and they're mostly present in the endosperm. Okay, so in terms of other specifics, if we talk about prolamins and glutelins, they have high amount of glutamine. About 30 to 40% of the protein is glutamine. High amount of proline, a low amount of charged amino acids. So very low amount of basic and acidic amino acids. Therefore, they're not soluble. It's very hard, like at pH, at neutral pH, they're not soluble. So they're not soluble in water. They have considerably low amount of cysteine, but they are present mostly in the high molecular weight prolamins or glutenins. But these are located strategically at the terminus of the glutenin molecules. That means at the N-terminal and C-terminal, you have a concentrated amount of cysteines, which is convenient. Then you can form the disulfide linkages between the different molecules during kneading of the flour. So glidin is mostly monomeric, so basically it does not form a quaternary structure by itself. It has some cysteine residues, but mostly in the alpha, beta, and gamma gliadins. There's the omega gliadin that lacks cysteine residues, but all of the cysteine residues in the alpha, beta, and gamma are involved in intra-chain disulfide bonds. That means they don't form disulfide bonds with each other, that hence they're monomeric, but they have disulfide linkages within the molecule itself. And it's mostly responsible for the viscosity and extensibility of the dough or the gluten in the dough. So hence the viscoelastic properties. So glidin is mostly responsible for extensibility and viscosity, And the glutenin, on the other hand, is responsible for elasticity. So if you have a high gliadin to glutenin ratio, then you have a more extensible dough than elastic dough. Yeah. So the glutenin, in contrast to gliadin, is polymeric. So it forms a polymer, quaternary structure, mostly by disulfide interchange. So each monomer is around 70 to 90 kilodalton. And we do have two classes. We have the high molecular weight glutenin and the low molecular weight glutenin, but they're linked by disulfide linkages. So under reducing conditions, when we run the gel in reducing conditions, you can see them separate. Under non-reducing conditions, you see them migrating together. And as I said a minute ago, high molecular weight glutenins are mostly responsible for the elasticity of the dough. So the more proportion you have of glutenin in the flour, the more elastic is the dough is going to be. So glutenins consist of three domains. So we have the central domain, and then you have the terminus. So the central domain is made up of repetitive units. So certain amino acid sequences that repeat. And whereas the N-terminus and C-terminal ends, they're not repetitive. That means the units do not repeat. The amino acid sequence does not repeat. However, they're rich in the cysteine. And that's what facilitates the protein-protein interactions. There is a single cysteine residue in the middle domain of some high molecular weight glutenin subunits. So this is if you run the gluten proteins on a gel. This is what you see here. So this is under reducing conditions so that we can break up the high molecular weight glutenins. So you have these high molecular weight glutenins up here. You have some low molecular weight glutenins also. And you have omega gliadins and then the alpha and gamma gliadins down here, alpha, beta, and gamma gliadins down here. So the gluten matrix is made up of gliadins and glutenins. They interact with each other upon kneading your flour with water. So when you're kneading the flour with water, the proteins denature, and then you start having interactions between glutenin and gliadin, not just within glutenin alone. So the gliadins also open up and you start seeing disulfide interchange, and then the sulfider groups that forming disulfide linkages within the gliadin break and form disulfide linkages with other glutenins and gliadins, and then you get these interactions here and formation of beta sheet. So this is just further illustration. So gluten proteins are insoluble at pHs between 6 and 9 due to their low concentration of acidic and basic amino acids. However, if we reduce the pH or increase the pH above and or below that range, then they become slightly more soluble. When we're kneading the flour with the water, what happens also is changes in the secondary structure where we form beta sheet within the strands of the proteins, not just among the strands of the proteins from different molecules. So they come together and they form hydrogen bonding in the form of beta sheet structure. And this beta sheet structure is very critical in terms of the elasticity of the dough and the stability of the dough. And the formation of hydrogen bonding is the driving force for disulfide interchange. in a sense that it brings the strands in closer proximity to each other and facilitates the proximity that is needed for disulfide interchange. And the reason we have a lot of hydrogen bonding is due to the high glutamine content. So the high glutamine content in these proteins facilitate hydrogen bonding between those different molecules. So once they're formed, mostly by beta sheet formation through hydrogen bonding and disulfide interchange, you get that unique characteristic network that is a viscoelastic network, and it's insoluble in water. There has been research done over the past 2 decades that basically look at another form of interaction that also is important in building that structure, and it is dityrosine or tyrosine cross-linking. So this paper here, published a couple of decades ago, looked at the formation of tyrosine cross-linking, and they observed this using advanced LC-MS analysis to determine the presence of dimers and trimers of these tyrosine linked proteins. So, and they found that the tyrosine cross-linking is enhanced in the presence of oxidizing agents such as like potassium bromate. It enhances the formation, and while a reducing agent like cysteine counters that formation, the cross-linking. Also, an oxidative enzyme like peroxidase, they found that it would actually enhance the formation of radicals, like this is a tyrosine radical and another tyrosine radical, they are formed upon oxidation with peroxidase, and they form dityrosine crosslink. So this research had found that the stability of dough is not only due to hydrogen bonding and disulfide linkages, but also due to the tyrosine linkages as well by oxidation and oxidative reagents added. So this is an illustration of the bonding. So you have the hydration, and then you have the open up of the protein structure, and then you have the formation of hydrogen bond, disulfide interchange, and the tyrosine linkages as well. So let's go back a little bit to the role of glass transition. So the role of glass transition, here's a question for you to think about based on what we said at the beginning. In order for the protein network to form, you need the protein to be flexible. So it needs to go from a glassy state to an amorphous state. So what enhances that is presence of water and temperature. So for gluten in particular, like I said earlier, you would want to have, you can form the dough at room temperature at a particular moisture content. So for example, 13% moisture would give you, at room temperature, you can form gluten. But you can form it at lower temperature if we add more water. Yeah. So that is important in the formation of gluten. We have to be above the glass transition temperature. So like I said, formation of beta sheet is really important for maintaining the gluten structure and stability. So forming the hydrogen bonding among the high molecular weight subunits, and due to the presence of the glutamine residue. And when you put the flour in water and start kneading, it's sticky, right? So the water is not yet absorbed very well. So with the protein. So as with the more kneading you do, water gets really absorbed with the protein. And as the gluten network starts to form, it interacts via hydrogen bonding with each other and also with water and contains the water, then the stickiness. If you're in the kitchen and you know what I'm talking about is when you put the water, it's very sticky at first and then you continue kneading, and then the water gets absorbed and then you lose the stickiness. But if you continue to knead for more, like then you have to, after you reach this balance and you continue mixing, then you break down the structure and then you lose more, you start losing moisture, and the moisture comes out of the matrix and then gives you stickiness again. So because you're breaking now the structure. So the dough, the wheat-based dough is very characteristic again in the sense that you have the elasticity and you have also extensibility. So you have a nice balance. You can extend the dough, but it doesn't break very easily, and it maintains its strength, a coherence. And it can, once it's fermented, you have the dough rises and it can contain the gas bubbles or the air bubbles that is generated during fermentation. If it's a weak dough system, the air bubbles are not contained, and then you have a collapse of the system, and you don't have a nice rise. So if you want to make a loaf of bread with a nice volume, you don't have a nice volume if you have a weak dough system. If you have a weak gluten network, then you don't hold gas, and it's not elastic enough to hold the gas and then not collapse. So you won't have a nice volume if you don't have a good balance of viscoelasticity there. So the way we measure the viscoelastic properties of the dough is by a texture analyzer and a Kiefer rig. That's one way of measuring rheological properties. There are many ways. Farinograph is one way of measuring the rheology of the dough. But here, if I want to determine extensibility and strength of the dough, there's this Kiefer rig that will basically you prepare strands of dough, and then you would pull up the strand, and then you'll measure the height, how much you can pull it up before it breaks. So that's a measure of extensibility. And the force is the measure of elasticity. The more force you need to pull it up, the more elastic is the dough. So here are some questions for you that might come in an exam or might be in your practice set problems. How would reagents such as reducing agents, agent that promotes disulfide linkages like oxidizing agents, and thiol blockers affect dough structure? So reducing agents basically can reduce the disulfide interchange, disulfide linkages, so might enhance extensibility and reduce elasticity. So if you have a flour with a very high ratio of glutenin to gliadin, like you have high molecular weight filaments or glutenins, so you don't want, it's too elastic, you want some viscosity, some flexibility and extensibility. So you can add a reducing agent or a thiol blocker that blocks the sulfidyl groups. So that can reduce the strength of your wheat gluten. Or you can add oxidizing agent if you want to increase the strength of the gluten, and you have probably higher gliadin to glutenin ratio. So in order to counter that, you can add ascorbic acid, for example. So you can enhance the elastic properties of the dough. So that brings me to the gliadin to glutenin ratio, which we talked about. The higher the gliadin to gluten ratio, you have more extensibility and less elasticity, and the opposite is true. And changes in the amount of high sulfur containing gliadins. So you have the omega gliadins that is lacking in sulfidyl group, and then you have the alpha, beta, and gamma gliadins that are high in sulfur group. So although they are monomeric, but in their presence and the amount of sulfur or sulfidyl groups in them enhances protein-protein interactions during the gluten formation. So a higher proportion of the high sulfur containing gliadins also enhances the strength of the dough. So this is a very classic article or research done on gluten to show you the differences between gliadin and gluten in terms of viscosity and elasticity. So here in this picture, in the left side is gluten. Basically, it has gliadin and glutenin, so it has elasticity and it has extensibility characteristic. This is a gliadin fraction alone. So if we're trying to mix gliadin with water, you really won't get an elastic structure because of the lack of the high molecular weight prolamins or glutenins. So you get really a viscous and very extensible type of dough. If you only have glutenin and you don't have any gliadin, you hardly have any extensibility. So it's very strong, very rough, elastic network. So the way they found this is by fractionating and reconstituting. So they fractionated the protein within the wheat flour into different components, and then they mixed them back at different proportions and determined the impact of the ratio on the viscoelastic properties of dough. So studies that work on, that look into the role of the gliadin to glutenin ratio starts with fractionating, removing the protein from the flour. And this is the most traditional way of doing that, which actually I did in my master's degree research, is I took the flour, I defatted the flour, removed the oil, and then I made a dough. And from the dough, I washed the dough under water and saved the starch component in the water, because the starch should be washed with the water, and then what remains in my hand is the gluten network. So I keep washing it until there is no more, the water is clear that comes out. So I take the water with the starch, I freeze dry that, so I have the starch, I have the oil, and then now I have the protein. And the protein is further fractionated into gliadins and glutenins. And then what I did is I took each fraction and then mixed it at different proportion, different ratios, and then added the oil and the starch and formed the flour again and baked into bread and determined the ratio. At the time, I was looking at flatbread or pita bread. Determine the ratio of gliadin to gluten ratio and the impact of the differences in ratio and the baking properties of pita bread. So, but there's another way of fractionating them. You don't have to make the dough and you don't have to wash with water. What you can do is you basically get the defatted flour, or if it's not defatted, you defat it, remove the oil, and then you extract with water and salt or salt solution. You can remove the albumins and the globulins. And you centrifuge basically the supernatant, the albumin and the globulin, and then the precipitate has whatever residual fiber, starch, and protein. So then you want to solubilize the protein. So there you add your alcohol, isopropanol, a reducing agent, and acetic acid. So now in this mixture here, you're solubilizing the gliadins and you're solubilizing the glutenins. So the prolamins, basically, the low molecular weight and the high molecular weight prolamins. And you centrifuge, and the precipitate is mostly starch and fiber, and then in the supernatant are your glutenin and gliadins. If you want to separate the gliadins from glutenins, then you do the extraction here with only isopropanol, and that will solubilize the low molecular weight prolamins, which are the gliadins. And then you take the precipitate, and then you add to the precipitate the propanol again, but with BME and acetic acid. So now you're solubilizing the high molecular weight prolamins or the glutenins. And then you can get gliadins separate from glutenins. And this is if you're running the gluten mixture here after all this isolation, this is what you see on the gel. The high molecular weight glutenins, the gliadins, and the low molecular weight glutenins. So again, the ratio impacts the bread quality, whether if it's elastic enough or strong enough, you have a higher loaf volume. If you want to produce pita bread, you want more extensibility than elasticity. You want to flatten that bread and you don't want it to shrink. So basically you want a type of flour that has a higher gliadin to glutenin ratio, in contrast to the loaf bread type of bread. Just a little bit on zein, which is corn protein. Zein is the most, alpha-zein is the most common protein in corn, and it's very hydrophobic. So it's not soluble at all, and it has much higher hydrophobicity than gluten itself. And it doesn't function, it doesn't have the viscoelastic properties that the gluten-forming protein has, because the prolamins in zein or alpha-zein is a prolamin of lower molecular weight. It doesn't have glutalins, so you don't have the high molecular weight prolamin. So you might be able to form a viscoelastic protein network, but you need to have a higher temperature. So you have a higher glass transition. So that protein has needs higher temperature to change from glass stage to amorphous state. So this is basically, the glass transition is about 28 degrees C at a moisture content that is higher than what you need for the wheat protein. So you can form a viscoelastic dough at 35 degrees C. However, zein lacks the high molecular weight prolamins or glutenins. And what happens to the structure is what you see here in this image, it crumbles. So you lose, so this research here, Mea et al., what they did is they looked at the secondary structure of the dough at certain, after formation and when they let it rest. So they noticed that the beta sheet formation is not stable over time. So you lose beta sheet over time. So that's an indication of instability of the network. And therefore, when you bake it, it just crumbles and does not hold the gas. So it basically collapses due to unordered structure. Ways to enhance bread from corn is adding hydrocolloids like the hydroxypropylmethylcellulose can enhance the structural properties of the bread. Or if we want to formulate, in this case, is rice, also it is low in high molecular weight prolamins. We can use transglutaminase, which is an enzyme that promotes covalent linkages between the amine group of lysine here and the carboxyamide group of glutamine. So this is here is a research that we've done in the lab. We looked at a grain called intermediate wheatgrass. So this is related to wheat, but it's a perennial winter grain that has environmental benefits. But when we looked at its characteristics for application in like bread, for example, it has had poor performance. And when we tried to look at reasons behind that, so we extracted the protein and we looked at the composition in comparison to wheat. So here in lane 2, you have the gluten forming proteins. And then here are different lines of intermediate wheatgrass. You can see that there is the high molecular weight glutenins are lacking, or there are some, but they are deficient compared to those present in wheat. And we see a lot of the alpha and gamma gliadins. So you really have very high gliadins to gluten ratio. But they are not gluten-free. So if we want to formulate with them, we cannot say it's gluten-free because they have some of those characteristic components. Gliadin is also responsible for some allergenicity and sensitivity, gluten sensitivity. So ELISA showed that it is positive in terms of comparison to gluten-forming proteins in wheat. So we tried to look at characteristics of the protein to determine, other than it's the presence of higher gliadin to gluten ratio, we looked to see at the formation of beta sheet and secondary structure in a dough. So we did FTIR and we looked at the secondary structure, and this here on the left is the flour, and then here on the right is the dough, and you can see this huge peak here due to the water absorbs in FTIR. So we did correct for the water and deconvoluted the peak, the amide 1 peak, and we found that intermediate wheatgrass has lower beta sheet than wheat. So here you have 0% intermediate wheatgrass and 100% wheat. And this is, in this case here, is 100% intermediate wheatgrass, 0% wheat, and then these are in blends of both. So we found that the dough has less beta sheet, a little bit more random coil, which kind of describe or also explain the low stability of the viscoelastic structure in this system. And when we did a deep dive into the percentages of gliadins and glutelins, which is high molecular weight polymeric protein, in hard red wheat, we have a higher percentage of high molecular weight protein compared to intermediate wheatgrass. Forget about all of these are different lines, but we have a higher percentage of high molecular weight polymeric protein, mostly due to glutenins, and we have slightly less gliadins, and we have slightly less albumins and globulins. So these are the non-functional proteins, or the proteins that do not contribute to viscoelasticity. Doing a farinograph, we see also a characteristically weak dough formed compared to hard red wheat, and also at and when it hydrates and when you mix it, you reach dough formation quicker, so the dough forms quickly and then also breaks quicker, so it kind of sticky mess does not develop very well. So these are different breads with whole wheat flour versus intermediate wheatgrass. So they're denser. You don't have the elasticity to hold the air, so they kind of collapse, and the dough doesn't rise very well, so don't get a nice low volume. We do get cookies. You can get good cookies with formulations, a lot of butter and sugar. You get a nice cookie, it doesn't need elasticity, it just works out well. And that's it. So this is it for cereal proteins. What would be left? The topic that is left is protein modification, and we'll cover that on the 21st and finish it on the 28th. Okay, see you all later.